Tekrardan merhaba herkese. Hepiniz hoş geldiniz. Okay then. Good afternoon to everyone, and welcome to another session of ISCB RSG-Türkiye's webinar series. First of all, extend our greetings to all biologists on World Biologist Day. Today's will be given by Professor Dr. Tunca Doğan from Hacettepe University. The subject of talk is Artificial Intelligence Driving Approaches for Biomolecular Representation, Property Prediction and Design. Let me give you some information about Professor Dr. Doğan before we start. Professor Dr. Tunca Doğan was a postdoctoral researcher at the UniProt Protein Database team at UK's European Bioinformatics Institute and Cambridge University, working on computational protein science. Professor Dr. Doğan is a faculty member at the Department of Computer Science and AI Engineering at Hacettepe University, Turkey. Also, he is principal investigator of Hacettepe Biological Data Science Laboratory. After the talk, we will have a question and answer session. So if you have any question, please use the chat box. Now, I would like to call upon Professor Dr. Doğan to give his talk. Thank you.
Thank you very much, Ayşe, and welcome all. It's a great pleasure to be with you today. I would like to start by thanking RSG-Türkiye for organizing this event. Let me start by sharing my screen. Okay, now I'm going to make it.
Recording in progress.
Okay, I guess it's now sharing, right? You can see my screen?
Yes, we can see it.
Okay, perfect. Then I'm going to start. Today I'm going to talk about a few of our projects that we've been conducting in the last 2 years, we can say, 2.5 years. But I want to spend some time for the question and answering session as well, because otherwise it would be more like a lecture, so I don't like that. I want this to be an interactive session so that you can ask questions about the research that I'm going to talk about, or something unrelated in general in the field of, about the fields of bioinformatics, cheminformatics, or AI, anything you can ask, then we can, continue as a chat. So let me start by talking about the lab focus and projects. We are the Hacettepe University Biological Data Science Lab, and we are trying to develop and apply some artificial intelligence-centric approaches to biological and biomedical data. And the focus is usually on proteins, but sometimes we deal with small molecules as well. And the aim is building and testing hypotheses about life. So we are mainly developing new methods, computational methods, and then deploying them for other researchers to use. Other bioinformaticians, they can be molecular biologists or clinicians, can use our methods to further their own research. To do that, we have 4 different topics: computational drug discovery, systems biology, molecular design and embeddings using generative artificial intelligence, and predicting the functions of genes and proteins, we can say biomolecules in general. So for each topic, we have different projects that has been finished or still going on, but today I'm going to talk about 2 of them. And these projects that I'm going to talk about is centered on drug discovery and development. So in order to give some information about the projects, first we need to talk about what's drug discovery and development and what are the problems associated with them. Actually, discovering novel drug candidate molecules is one of the most fundamental and critical steps in drug development procedure. And it's a long procedure. It's taking on average 13, 14 years for the identification of a target, and it's usually a protein in our body, to treat a disease, and then constructing a chemical library and doing some screening experiments to see which of the molecules in that library can efficiently and specifically bind our target protein or proteins. There can be multiple of them, and then selecting the best options, best candidates, and optimize them. This is the job of medicinal chemists. And then there comes the clinical trials, preclinical steps on like animal models, and then the clinical trials on human. And if everything goes as planned, then the drug will be approved after many years. And the cost is also very high, nearly 1.8 billion US dollars. So the efficiency of this procedure is low. As you can see, it takes a long time, so the cost is high. So we try to modify these processes from different aspects, from different layers, in order to make it more efficient, okay? Reduce the cost, reduce the time, and increase the safety of these drugs in the end. And machine learning-based prediction models are used for this procedure, especially the procedure called virtual screening, in which we take a large chemical library composed of thousands, sometimes millions of drug candidate molecules and our target protein, and we try to see if these two will be interacting with each other. Normally, this procedure is done by physics-based algorithms like molecular ducking or molecular dynamics simulations. But since they took a lot of time still, even though they are computational, machine learning started to be used. And the aim is that which molecule, as you see here in the right-hand side of the figure, would bind specifically to the binding pocket of that biomolecule, so that the necessary modulation of the protein can be made. And to do that, machine learning models, as I said, can be used. Since machine learning is a data-centric approach, you need known data with labels, known labels. So which molecules are known to interact with which proteins. By getting this data from open access repositories such as PubChem, Campbell, and DrugBank, we can build our training and test data sets on which we can train our model and then test the model, optimize the model, then use that optimized model to predict new interactions where we don't have any information about a target without any known drugs, or a drug candidate molecule without any known binders, without any known targets. We can test them together in the model and infer whether they are going to be, binding each other or not. But of course, this is not the only part of drug discovery. We also have different steps, like designing a drug, for example. This is another important step. Normally, this is done manually by, again, medicinal chemists, but the procedure is, again, not very efficient. And another important part here is that knowing the properties, physicochemical and biological properties of those molecules. If you know this before the animal models and clinical trials, then you can make a better judgment about which kind of molecules could become drugs at the end of the procedure, so that you can increase the safety of the drug candidate molecules. And another important thing is that since most of the cost is associated with these clinical trials, if you can predict the outcome of this trial, then you would make this procedure much more efficient. And for each one, we had some projects in the past, and we developed some methods that we call deep screen,
Recording
cell formers, or drug app. These are the names that we give
Recording
to our
in
models
progress.
that are realizing these aims, such as the drug approval prediction, or the predicting the molecular properties, physical chemical properties of these drug candidate molecules. Each is trying to contribute to the field of drug discovery from a different perspective. And today I'm going to talk about two of these projects. The one that we developed for designing new drugs automatically using AI, DrugCan, and for predicting the molecular properties of those designed molecules in the road to become drugs, called cell formers. So let's start with the first one. The title of the project is De Novo Design of Drug Candidate Molecules with Graph Generative Deep Adversarial Networks. As I said, it's important to predict the unknown interactions between drug candidate small molecules recorded in chemistry databases. However, their number is limited in the scale of hundreds of millions. In the PubChem database, we have nearly 77 or 80 million drug candidate molecules. In Campbell, we have 2 million drug candidate molecules. And in on-demand chemical libraries that is used by pharmaceutical companies, the number is at the scale of billions, 1 billion, 2 billion. And the largest one is, as far as I know, is like 30 billion. However, on the other hand, it's possible to design approximately 10 to the power 60 small molecules different from each other by at least one atom or bond, indicating the nearly limitless potential of theoretical space of drug-like molecules. 10 to the power 60 is a vast number, actually. It's not like a 10 billion or 100 billion. So you can imagine how large this number is by giving an example. For example, the total number of atoms that forms our planet Earth is in the scale of 10 to the power 55, 54 or 55. This is an estimate given by physicists. And the theoretical number of small molecule drugs that we can synthesize in theory is nearly 1 million times higher than the number of atoms that form our planet. So it's a vast number. And normally, since we don't know these molecules have never been synthesized artificially or have never been extracted from nature and never existed in nature, most of them. And it's possible to record them and then try to synthesize them. Not all of them will be stable molecules that can exist in our universe because of the physical laws, physical constraints, but many good candidates, drug candidates can be obtained from there. And since it's not possible to design and synthesize one by one, these 10 to the power 60 molecules, one good option, alternative, is using generative deep learning. Generative deep learning models create new data points according to a probability distribution at hand. The probability distribution at hand is our training data, which are known drugs in this case. And these models have been developed with the purpose of picking completely new samples from a distribution space that is only partially known. So this has been utilized in many different areas up to today. For example, these deep fakes, which are human images that are not real, completely synthetic, generated by the model. And the first model that has been developed was nearly 10 years ago, in 2014. These models were successful in generating human faces, but as the years passed, the models and technology developed, and in 2018, 2019, the images generated by these models, it's not possible to differentiate them from real people's images, real people's photos. And today, they are even much more successful. And today, it's even possible to generate completely new videos, let alone images. And another type of generative modeling approach is the text-based generation. As you all know, ChatGPT, OpenAI's GPT-4 model, and other language models, they are using architecture, a deep learning architecture called the transformer architecture. And most of the models that generate these synthetic images were using generative adversarial networks. Today, diffusion models are, more successful, but these architectures can be combined with each other to get a better result. So the idea in this project is to using this generative modeling approach and these architectures in order to generate target-specific, protein-specific molecules, and design these molecules automatically, and then we can use those molecules in the later steps of the drug discovery procedure to come up with better candidates, drug candidates. The main aim of this project is actually coming up with this model, this AI model, to automatically design target-specific de novo molecules. And the model that we developed here, that we call DrugGAN, explores the recent and highly successful architectures used in many different fields, such as the natural language processing, computer vision, or network analysis. And these are the transformers, graph neural networks, or generative adversarial networks. And when you develop an AI model, of course, you need to test it on a real world case to see if the results produced has a biological meaning or not. And the first application of our system as part of this project is coming up with effective drug candidate molecules for hepatocellular carcinoma disease, which is a deadly subtype of liver cancer. And in this disease, a signaling pathway called PI3K-AKT-mTOR is hyperactive. And a key protein in this pathway is ACT1, 2, and 3. There are 3 isoforms of this protein, ACT proteins, we can say. They have a key role in this pathway so that coming up with a specific binder that target the ACT protein is important. And still the research, this is an active area of research. Many researchers has been working on to develop a specific and effective inhibitor for the ACT protein. So we try to design some molecules to interact with the ACT protein, and then the aim is to validate our results by first synthesizing these new de novo molecules that is created by our model, and then validate their activity against this protein in the hepatocellular carcinoma disease in vitro lab experiments. And in the end, the aim is to construct the system as an online service so that the researchers can run these models to design their own de novo molecules for the target of interest. The workflow is summarized in 3 steps that you see with 3 different colors. The orange one is about the gathering and organizing the data sets. We get our small molecules, targets, and bioactive molecules, the bioactivities, the interactions, known interactions, experimental interactions between the targets and small molecules from the Campbell database. And for proteins, we use the UniProt and PDB. And then we prepare our data sets and then change their format so that the model can take them as input. For that, we are using the graph data structure, and we encode our molecules using graphs. And then we take these molecules and then train our model. We come up with a model based on generative adversarial networks. I'm going to explain them in detail in the following slides. And use the data sets that we have gathered in the previous step to train the model. And then in the end, the model produces completely new molecules. And then we take those molecules and subject them to benchmarking experiments to compare the performance of the model with other models from the literature. And then we also test them to see their drug likeness, to judge would it be possible to develop some drugs out of these molecules created by our system. And in the last part, where you see at the bottom of the slide with the red color, we take many molecules, thousands of molecules created by our model, and then we subject them to filtering operations. First, drug likeness filtering, and then we see their physicochemical property distributions and compare them with the real drug. And we visualize our distributions using the dimension reduction approach, and then use some physics-based approach, such as molecular docking, to see whether these molecules can really bind to the intended target, ACT1 protein. And we also do, in parallel, bioactivity prediction. This is similar as molecular docking, but from the machine learning perspective. And then we get our results and filter our molecules, reduce their number to a reasonable degree. And then in the scale of hundreds, we use our project partner experts so that they can eliminate the remaining ones to come up with the most promising molecules. And we select 1 or 2 and then use molecular dynamic simulations to see the temporal binding properties of the de novo molecules against the intended target to evaluate our model's results. So it's a quite comprehensive elimination and selection procedure that's going on after the training and testing of the model. So as I said, the starting point is the data gathering step. These generative models are data hungry, so you need to use a large data set. And to train the DrugCam model, we use the Campbell molecules, and we come up with some filtering operations to only get the drug-like molecules and eliminate the rest, because the out of distribution training samples can make the model worse. And our target protein is, as I said, ACT1 protein, and the size distribution is shown in this histogram, where we take at the highest level 45 heavy atoms as our limitation, because larger than that the model should be really complicated and learning is not easy. So once our data is ready, we come up with our model. We use the PyTorch library. These are deep learning libraries, are encoded, prepared by professionals, so that the models can run in an optimized manner. So we are using PyTorch and then their functions. But of course, we come up with our own model. Actually, this was the first time, as far as we are aware, where a language modeling architecture transformer has been placed inside the generator of the generator adversarial network, in which the graph-based data is processed. And here how it works, let's see. The input, which is actually the noise, or real molecules can be used, both is possible, has been subject to some linear layers in which the weights of the model are optimized in order to learn the patterns in the real drug molecules, or drug candidate molecules, we can say. The transformer encoder is actually trying to learn this by using the attention module. The attention module tries to find which parts, which atoms and bonds are important in scale to the other atoms and bonds in the same molecule. And once this learning procedure is going on, at each step, a new de novo generated molecule has been produced. And it has been given to the discriminator network, which is a second network, again composed of linear layers. It tries to predict whether the molecules coming from the generator module are real and fake. Of course, the ones coming from the generator are fake, but the discriminator does not know this, because at the same time, we give real molecules that we get from these databases, such as Campbell and DrugBank, together without giving any labels, so that the discriminator is going to see sometimes the de novo generated molecules, and in other times real molecules, without knowing which is which. And then it tries to predict as real and fake. If this discriminator is successful, then the result is feed back to the generator to tell the generator module to work harder to produce a better molecule that resembles the real drugs. And if the generator is really successful, then this time discriminator starts to fail. So there's a zero-sum game going on between these two modules. They try to learn from each other's mistakes and do better at each iteration. And in the end, at the end of a long training procedure, both is really good at their job, so that the important part is, of course, making the generator better. So that at the end of the training procedure, the aim is that generator is producing molecules that are original, not the same as the ones in the training data set, of course, but at the same time, they are drug-like and have high potential to bind to the intended target, which is the ACT1 protein in our case. And when we trained our models, we see that these molecules has been produced. These are molecule record that has never been synthesized before. These are original molecules. And it's interesting to see that these molecules have never existed in nature or in any database before. These are the results of this model. And then at this point, we see that, okay, the model is able to produce realistic molecules. Now, let's go to the next step and optimize the model further to see how it behaves in terms of producing targeted molecules, the molecules that are able to target our intended protein. And after that, using our optimized models, we did a performance analysis, because in a method development study in machine learning, especially in machine learning, you need to compare your model's performance against others that are doing similar jobs in the literature. And for the molecule generation purpose, there are two main types of methods. The models that are using text, taking text as input, like the natural language processing models, and the ones that take graphs as inputs and produce a molecule in the graph data structure. And DRAGGAN is a graph-based model. And then we compare our model's performance against others. One of the comparison is made over validity. This is a metric that measures how many of your molecules are valid, real molecules that can exist in when synthesized. Of course, a higher number is better here. And as we see, the DRAGGAN models is not perfect in that sense, because the one is the perfect result. However, the important part here is that there is a trade-off between validity and our second metric, which is novelty. Novelty is about producing a new molecule that does not exist in your training data set. This is also very important, because if your model is producing a valid molecule, but the same molecule that already exists in your training data set, that model has not any real use. This is just producing the same thing over and over again. So that the aim of training a generative model is coming up with these novel molecules, new molecules, okay, that we did not have before. But there is a trade-off between validity and novelty. If you want to increase novelty, the validity decreases. If you want to increase validity too much, then novelty can decrease. And finding a good balance in between is important. And DRAGGAN performed quite well in terms of novelty and not bad in validity. There are other metrics as well, such as uniqueness. So how many of the generated molecules are different from each other? So the comparison is not between de novo generated molecules and the training data set, but the de novo molecules generated by this model, specific model, are compared within each other. Uniqueness should be high as well. And internal diversity is a similar metric, like uniqueness. And CAT is the estimated score for drug likeness. It's also better if you have it higher, so that we find that our model is performing better than others in some metrics, worse than others in other metrics, but in overall it performs well. But a performance comparison is not sufficient to evaluate a new model, a new machine learning model. So what should you do is that you need to explore your results from different angles, from different perspectives. And one of the things that we did here in this project is first we subject them to the classical filters, drug likeness filters, like the Lipinski or Weber or Payne's filters. And we see that nearly 50% of our molecules are suitable, that can become drugs, that have the properties of the drugs that we have today, which is not a bad number. And another thing that we did is the visualization using dimensional reduction techniques. Dimensional reduction is an unsupervised learning technique in machine learning, where you are able to visualize your results, to see the groupings and judge on your results. So here at the top, what you see is that there are a lot of dots. It's quite crowded, but each dot is representing a molecule. And the blue colored ones are actually the molecules, the novel molecules created by our model. The gray colored ones are randomly selected molecules from our training data set. These are real molecules that represents the whole drug-like molecule space. And the yellow ones are the known real inhibitors of this protein, ACT1 protein. So what we want to see here is that on this distribution, if our blue molecules, de novo molecules, are close to the yellow molecules, then it means that we can infer that, our de novo molecules are carrying the properties of the real ACT1 inhibitors, which is a good thing, of course. And at the right-hand side, we did the same analysis, and we see that our blue are close to the yellow ones, the real ones, which is again a good thing. In this one, we use another dimensional reduction algorithm called TSNE. I'm not going to go into detail of how these algorithms work, but if two molecules, two dots are close to each other in this two-dimensional space, then it means that their properties are probably similar as well. So that blue is close to yellow means that our de novo molecules are carrying the properties of real ACT1 inhibitors, so they have high potential to inhibit the ACT1 protein. At the C panel, what we see is the result of the molecular docking experiments. Another type of validation that we did. Molecular docking is a physics-based method, so you can validate your machine learning methods from another perspective using this physics-based algorithm. We take our de novo molecules and dock them against the structure of the ACT1 protein, and we also did the same using the experimentally known real molecules, real inhibitors of the ACT1 protein, again shown by this box where inside the yellow color exists. And here, if the box is close to the top of the y-axis of the plot, it's better results. Okay, the binding affinity is negative, and -10, -11, -9 are good results. Again, I don't have time to go into detail, but we want the box close to the top. And as we see for the drug can, the box is around -8, -9, and similar for the ACT1 inhibitors, which shows that the real ACT1 inhibitors, which are experimentally known, are good, but our de novo molecules are at least as good as them, in some cases better. Again, we did another experiment using machine learning, trying to predict bioactive molecules. From our de novo molecules, again created by DrugCan, we subjected to our previously developed system, deep screen system, to predict them as active or inactive against the ACT1 protein. And again, we see that more than half of them has been predicted as active, which is a good result as well. All of these results are trying to validate the outcome of the model to see whether the model works well or not. And then, as I described in the one of the first slides, we are filtering molecules at each of these steps. For example, if the molecular docking experiment result is bad, we eliminate those molecules. If deep screen system is predicted a de novo molecule as inactive against ACT1, we eliminate them, and then we reduce the number. We start by producing 10,000 de novo molecules, and then this is reduced to 5,000, 1,000, 100, and in the end, with expert curation, we come up with 25 candidate molecules, and we conduct molecular dynamics experiment on a selected showcase molecule, which is this molecule that has never existed in the literature before, first time created by this model. And we compare the molecular dynamics experiment results with the real known, experimentally known real molecule, real inhibitor of ACT1, and we see that the binding properties, the binding modes are really similar, which again verifies the result of the model. This is still an ongoing project. The next thing to do is actually synthesizing these molecules, which we are doing right now, and then trying them on in vitro experiments to see whether they are really inhibiting the ACT1 protein and also acting on the hepatocellular carcinoma cells, killing the cells or inhibit the cells at least. And if the expected results are achieved, it may be possible to potentially discover new drug candidate compounds that are critical for the treatment of hepatocellular carcinoma. But this is not the outcome of this project. Of course, we need to conduct new projects. This is an ultimate goal that can be achieved in upcoming years. And the source code, the data and trained models are all shared in our data repository at GitHub. And we also come up with a web-based DrugCan service with a graphical user interface. These screenshots of which are shown at the right-hand side of this figure. Here, you can go to this URL where you see at the left-hand side, and then by selecting the number of the molecules that you want to create and whether you want to target ACT1 protein or you just want to create random molecules without targeting any specific protein, you can start generating molecules and then download these new molecules to your own local machine. And then you can do whatever you like with them. Maybe you can use them in your own project. Today, we have a preprint article. Of course, it came in the late 2023, but we still have this. The new version is coming up, and it's also in peer review in a journal. I think, yeah, this is the end of the project. And at the end, I want to thank the project members. So this is an interdisciplinary project, and three different universities, teams from three different universities are working on this project. And for the AI and bioinformatics parts, we are doing as the Hacettepe University Biological Data Science Lab. And all of the students that take part is listed here. Atabey, Elif, Ahmet, Gökay, and Osman, Hayriye, and Dr. Ahmet Rifaioğlu and Heval taken part in developing of the DRAGCAM model and the data set and doing the downstream analysis. And for the pharmaceutical chemistry part, our collaborators from the Gazi University, Professor Dr. Erdem Manoğlu and Dr. Abdurrahman Olvaç. And for the cancer-based experiments, we are collaborating with the Cancer Systems Biology Lab in Middle East Technical University. Dr. Deniz Kahraman and Altay Koyaş and Müge has worked on this. And for funding, I want to thank TÜBİTAK, Bilim Akademisi, and TÜSEP for their support. This is the end of the first project. Now I want to spare some minutes. I want to be quicker for the second projects, in which we developed a new model called CellFormer, Molecular Representation Learning via Cell-Based Language Models. So what is this doing? Actually, this is trying to predict molecular properties. This model is predicting molecular properties of these drug candidate molecules. But what are them? Molecular properties play a crucial role in drug discovery, and understanding these properties helps in designing effective and safe drugs. So once you come up with a design, then you can use this model to predict their properties and maybe eliminate the molecule if the properties are not as desired. And key molecular properties determine a molecule's pharmacokinetics, pharmacodynamics, and safety profile. A few examples to these important molecular properties can be given as, for example, aqueous solubility of a molecule, or lipophilicity, or blood-brain barrier permeability, or their binding to specific biomolecular targets. Each one is quite important for them to be able to become drugs. Because, for example, you want your drug to be soluble in water, but this the magnitude of this solubility is important as well. However, another important thing is that it's difficult and expensive to identify these molecular properties of large chemical libraries because it's costly. You need to synthesize the molecule in large quantities and then subject them to different experiments and then get the result. Of course, you can do this with if you have 1 or 2 or 3 molecules, but imagine that you are trying to screen 1 million molecules. It will take forever and large amounts of money in order to conduct this. So that we are trying to do this computationally. Using atomic information of molecules, we can build machine learning models to predict their physicochemical or biological properties. And that's what we do here. And in order to do that, of course, we need to encode molecules, because molecule is something that exists in our universe. However, you need to encode it digitally to give it as an input to the model. Molecules are encoded and stored using alphanumeric characters to process manually, as you see here, for example. To store the atom and bond information, which is important for the properties of the molecule, we frequently use line notations. And one of the most widely known notations is called SMILES, Simplified Molecular Input Line Entry System. As you see here, there are different characters that are representing atoms. C is representing a carbon atom. And these other symbols are trying to represent the bond, whether there is a bond between two specific atoms and what type of bond is there. okay? Covalent bonds mostly. It's trying to encode this information in an alphanumeric line notation. SMILES is widely used, but it's problematic in different ways. For example, SMILES is not a unique way of encoding molecules, and also its strings may have invalid chemical properties, so that you need to make some assumptions while going from the actual molecule to creating its SMILES. And when you give the SMILES to the model, machine learning model, of course, it doesn't know your assumptions. So it doesn't know anything about the real state of the molecule. It only sees the SMILES notation so that it can learn the errors of this notation. So therefore, new notations are required in these fields. A new one called selfies, it offers 100% valid and robust way of writing molecules. And this was developed by a famous computational chemistry group from the University of Toronto a few years ago. And it's possible to develop computational models using selfies notations as the inputs. You can encode your molecules using the selfies notation and give these notations to the machine learning model and train a machine learning model to successfully predict the molecular properties that we've been talking about. And what we do in order to realize this is actually called molecular representation learning. In machine learning, representation learning, or we can call feature learning, is a set of techniques that allows a system to automatically discover numerical representations needed for feature detection. So these kind of models, for example, the transformer model, the transformer architecture that has been widely used in natural language processing models such as GPT-4, GPT-3, and others, are taking these alphanumeric representations like line notations, smile selfies notations, and natural language directly, the words of the sentences, and then they extract some numerical features. This is important because these numerical features, we can use them to make some predictions, whether they are molecular properties or other predictions as well. So how does it work? In order to understand it, we need to take a look at the natural versus molecular language modeling. Texts, as you see here, natural languages, proteins, and small molecules, they all can be represented as strings of letters and processed via these natural language processing methods to study their local and global properties. A language model is trained on self-supervised tasks over a huge corpus of unlabeled text. In this task called masked language modeling, some fraction of these words, for example, you have this text, and some of the words are masked. So they are not given to the model, and you expect the model to predict those words based on the context, based on the other words that has not been masked. So that if these words has been predicted successfully or not, will be based on the calculation of a loss value, which is fed back to the system through backpropagation, and the model is trained in epochs, so that in the end, at the end of the training procedure, the model learns how to predict these words that has been masked correctly. So in the end, when you have a successful language model, then you can use this model to different downstream purposes. You can fine-tune them on downstream tests over labeled data through a standard supervised learning approach. And we are using the same approach here used in natural language modeling to learn the chemical properties of these molecules. First, we come up with a foundational model, a pre-trained model, that we do not give any molecular properties. We just mask some of the atoms and bonds, and we want the model to predict those masked atoms and bonds correctly. And then when we have a successful pre-trained model, we use them, we fine-tune these models based on specific molecular properties. For example, predicting whether those molecules can penetrate blood-brain barrier by using against our benchmark data sets. So the aim of this model, aim of this project, we can say, is to integrate novel computational techniques, specifically NLP algorithms for representation learning, to efficiently explore the chemical space. So these are chemical language models. And for this, we developed Cellformer. It's a transformer-based chemical language model that utilizes self-is notation as input to generate compact and meaningful molecular representations for various stages of drug discovery and development. This model, Cellformer, seeks to overcome limitations observed in existing methods, and this is achieved by addressing the issues related to validity and robustness. And ultimately, the project aims to facilitate drug discovery and development by leveraging Cellformer to enhance the prediction of molecular properties in large chemical libraries. So you can give this model 1 million molecule or 1 billion molecule and quickly and successfully predict their aqueous solubility, okay, or potential side effects when they are used as drugs. In terms of the methodology, as I said, we first come up with a pre-trained model. This is an unsupervised training, or we can call self-supervised training. And in order to train this model, we use nearly 2 million molecules obtained from Campbell database. And then we tokenize the input using byte pair encoding, and we use the Roberta transformer architecture for this. We get the self-is notation and separate them into tokens, and we give it to the model. I'm not going to go into the technical details. The model learns the representation and then feeds out the numerical representation of the model. We come up two different models. Cellformer Lite is the smaller one, and regular Cellformer is our largest model. The large one, the main model, is having nearly 86 or 87 million parameters to optimize. Okay, and later, when we have a successful pre-trained foundational chemical language model, we fine-tune this model on specific molecular property prediction tasks. This is no more a self-supervised model. This is a supervised model. It becomes a supervised model. On top of our pre-trained foundation model, we place some classification, multi-label classification or regression heads, and then we give our benchmarking data sets. Each is a different data set, so we need to train a different model for each task, each molecular property task, and then train our model based on our smaller data set. So what if we didn't have any pre-trained model? We could come up with still a prediction model with this number of compounds. We could train a model. However, with this small number of compounds, the model would not be successful. So that the pre-training with a large corpus, unsupervised pre-training with millions of molecules, will make the model ready to be utilized in the fine-tuning procedure to come up with a successful model. And then after that, of course, after we come up with this model, okay, what we were saying here is that the pre-trained model, first, when you train a self-supervised model and then use it to fine-tune over a supervised task, for example, the predicting the aqueous solubility of the molecules in your data set, then it has a potential to become more successful. And we take this, our model, which we call Cellformer, and compare it with the other models in the literature. Again, we have two types of models, text-based models and graph-based models. In this project, we have a text-based model. Cellformer is a text-based model that is using the transformer architecture. And then we see the performance using the area under the curve, area under the receiver operating characteristic curve. The higher values are better, and 1 is the perfect prediction. Of course, this is a difficult task, and reaching 1 is not easy. And what we see is that for many molecular properties, our model performs well, not based on each of them. Sometimes graph-based models are behaving better, more advantageous for some of the molecular properties. And for other text-based models are working well. Here, what you see is BACE task, which is a protein, actually, human secretase 1. And the task here is predicting the binding properties of molecules, because in the data set, some of the molecules are binding to BACE, and some of them are not binding. And the second one is BBBP, which is the blood-brain barrier permeability, which is a very important property to develop drugs, especially if you are trying to develop a drug for targeting the central nervous system, because they should penetrate from blood to brain and then go and find the target and act on the targets. But there is a barrier in order to predict this, in order to protect the brain against invaders, foreign molecules in our blood that are coming from outside, so that a drug should be able to penetrate this barrier and correctly predicting the molecules that can penetrate this barrier is important. Another task is HIV, which corresponds to the ability of a molecule to inhibit HIV virus replication, binding to HIV viruses proteins and inhibiting the replication. And TOX21 is the quantitative toxicity measurement on 12 targets, whether a molecule is toxic or not. And CIDR, the last one, is representing the classification of drug side effects into 27 system organ classes. Again, the higher values are better. And the model, our model, Cellformer, is acting significantly better compared to other models in the literature for the CIDR task. Why? The question why is important here. It's most probably because that it's effectively learning the molecular properties that is related to side effects based on the molecular encodings that we use, self-is encodings. Actually, these were the classification-based molecular property prediction tasks, whether a molecule can bind to this target or not. We also have a real value prediction-based tasks, which are regression-based tasks. And the performance measure is error, root mean squared error. The lower is better. Perfect prediction is with error 0, but again, since these tasks are difficult, perfect prediction is not possible. And our model is the best model in the literature in terms of predicting aqueous solubility. The other two tasks are free sol is the hydration-free energy of small molecules in water, and lipophilicity is experimental octanol to water distribution coefficient of these compounds. And other models are acting better than our model. But the aqueous solubility prediction is realized best with Cellformer. So the results are discussed in a more comprehensive, better manner. And we also had some use cases, because use case analysis is important when you are developing a new computational methods, especially machine learning methods. So for that, we randomly select some molecules, and then we checked Cellformer predictions for blood-brain barrier permeability, also the side effect prediction, also the aqueous solubility prediction, and we check our results based on literature. We go and find the literature publications based on these molecules that is talking about these molecules and see whether our predictions are correct or not, and why. What's the reason behind this correct or incorrect prediction? This is for evaluating your model. This is quite important. And we also explore our data, again doing some dimensional reduction. In these plots, each dot is representing a different molecule. And then we reduce the dimensions. The original dimensions is higher, of course, should be 1024, I guess. And then we reduce them to 2 dimensions in order to be able to visualize. For the top, we see 2 colors, right? Blue and red. And this is done for the BACE protein binding tasks. The blue ones are active, binding to this protein, and red ones are inactive. And whether our model were able to separate these inactives against active ones, as you see this separation between 2 colors. If the grouping is really good, then it means that your model is successful in separation between these 2 groups of molecules. And as we see at the left-hand side, there is a clear separation, and the left-hand side plot is done by using the pre-trained model without any fine-tuning. So it shows that without even a fine-tuning on this task, the model were able to separate the inactive molecules from the active molecules. And at the right-hand side, the fine-tuned model's results are shown. Again, successful separation, and the separation in between these two groups are more linear, which means that fine-tuning even make the results better. However, the pre-trained model is acting quite good. And at the bottom, we did the same analysis for side effect prediction. Since we have 27 different side effects, we are using more than 2 colors. There are many colors. And again, the pre-trained model, only pre-trained foundation model, were able to separate the molecules in terms of their side effects pretty well. However, if you fine-tune the model on this data set, benchmark data set of CIDR, the separation is even clearer. And these discussions and everything, after everything, we share our ready-to-use programmatic tool together with the pre-trained and fine-tuned models, the representation vectors for all Campbell molecules, and everything in our GitHub repository. And in the end, we can say that this model exhibited high potential in molecular representation learning, and selfies-based representation models could serve as a powerful and persistent alternative to smiles-based chemical language models. And in the end, we published an article. If you are interested to see in more details, you can check our article, which was published in 2023, nearly 8-9 months ago, in Machine Learning Science and Technology Journal. We also have another publication, this time about protein language models. This is a chemical language model, but another project for protein language models, which has been published as, again, an article in another journal, Nature Machine Intelligence, in 2022. If you're interested in protein language models, then you can check that project as well, check that publication. And in the end, I would like to thank my students, Atakan, Erva, and Atabey, who worked on this project and developed the model. Now, this is the end of my presentation. And overall, I would like to thank my all lab members at Biological Data Science Lab at Hacettepe University, Department of Computer Science and Artificial Intelligence Engineering, we are composed of nearly 20 people, and these students are coming from different backgrounds. There are some computer scientists, there are some molecular biologists, or students coming from chemistry backgrounds or pharmaceutical backgrounds. Some of the students are from the master programs and PhD programs as well. And we have some undergraduate interns. The total number is, again, today is like 20 people in the lab. And also we have some partnerships, as I mentioned during the projects. We are working with Cancer Systems Biology Lab in Middle East Technical University and collaborating closely with European Bioinformatics Institute UniProt database team, and in Heidelberg University, Saiz Rodriguez Group. I would like to thank them as well for their cooperation, and all of my students. Thank you for your interest, and I can take some questions if you have time.
Thank you, Professor Doğan, for an interesting talk. If you have any questions, you can ask, you can write in the chat box. I see the chat box. There is 2 questions, actually.
Okay, if you can read so that everyone can hear the questions as well.
Okay, I can read it. Okay, Emre Bora says, can you share the GitHub link of those projects in here?
Yes, of course. The GitHub link for the second project that I talk about is here. Actually, the lab account is HU Biodata Lab. This is our lab account, and after that, of course, the project comes. This one is called Cellformer. The other one is DrugGen. So that's /Cellformer or /DrugGen. You can also find them if you go into the lab's GitHub page. So all of the repositories are listed there as well. The first project is called DrugGen. The second one is called Cellformer.
Another question is, maybe I missed it, but does your drug discovery model account for off-target effects, particularly for drugs targeting ATP binding sites, where specificity can be challenged?
Yes, actually, this is a good question. And as part of the project, we try to tackle this problem. You can tackle this problem in different ways. For example, the easier way to do this is you generate many molecules against your protein of interest, against your targets, and try to predict whether these molecules are interacting with off targets, the other targets, by predicting them. You can use molecular tagging, molecular dynamics, or machine learning based bioactivity prediction to see if they are interacting with other proteins. If they are, then you can eliminate those molecules and produce new de novo molecules from your model and keep doing this over and over. This is the easy way to do it. The more difficult but more rewarding way to do this is encoding this information in your AI model. Actually, you can encode this, and one of the aims of the project was doing this, but we still haven't come to that point yet. We try to encode this information so that the generated molecule during the generation procedure will not be generated against the other molecules, but only for the target of interest. And of course, if you can do this, this is more difficult to come up with a model like that. But if you can do this, then you don't have to generate thousands or millions of molecules and taste it and do it again and again in an iterative manner. You can just generate molecules, and you can be sure that they are not going to interact with other targets. But again, as I said, we are not at that point yet. I think there is another question, if you would like to read that as well.
Yes, I'll read. Thank you, Dr. Doğan, for this inspiring presentation. There is something I would like to know, a little bit off topic. I'm studying long non-coding RNA in hepatocellular carcinoma. Are there AI tools for cancer bioinformatics, biomedical bioinformatics, or RNA-seq analysis? Your suggestions are very important for me. Thank you.
Okay, thank you for the question. Actually, of course, there are many different AI-centric methods, or we can say machine learning methods to do this job. Not only one, there are many, and each is serving a different purpose or trying to, catch this problem from a different perspective. So that if you have your goal, if you have your problem that you want to solve specifically defined, and then you can go to the literature to see which methods are available for that purpose. Okay, maybe you can see for a specific problem, you can find 5 methods that came out as publications in late years, okay, in the last 4 or 5 years. And then you can compare the performance of those methods to see that which one would be better for your case. There are the performance comparison tables like the ones I shown during my presentation in those articles as well. You can use them. But at the same time, another important thing along with performance is the availability. Because in many papers, researchers are coming up with a tool with a good performance, but at the end, they do not release the code. They do not release the trained models so that it cannot be useful for the research community. We try to change this by, releasing each of our code, each of our method at the end of the procedure, but not everybody does this. But it's important for the open science. Actually, you can check whether the method is available or not. And if it is not available, you can just skip it. You don't have to waste your time on that. You don't have to check the performance. You don't have to read the publication altogether. If it is available, then you can check whether this method is performing well, whether it has the potential to solve your problem, and then you can select those models. Yes, I think this can answer your question. And depending on your problem, then the model, of course, changes. There is not only one solution. There are many possible solutions. You need to, consider these points to select a model and then try it. And sometimes you select more than one model and then use it on your data and see the results and compare the results on your own data to select one of them.
Is there any question? from audience. Oh, there is one more. No. Sugar says thanks a lot.
Okay, I guess there are no more questions. I'm going
Yes.
to stop sharing my screen.
Thank you for all, Professor. And I'm sorry for interruptions.
No problem.
Then have a nice day.
Okay, thank you very much for your interest. If you have questions, then you can email me as well. I will try to, reply those emails as much as I can. And
Thank
have a
you.
nice day. Thank you very much.